Okay, I think we should get going. So I want to welcome everyone to our Biostatistics seminar series. And I want to welcome our speaker, Dr. Jonathan Greenbaum, but in particular, I'd like to turn to one of our PhD students, Jessica Randazzo, who knows Dr. Greenbaum and invited him to give this talk to introduce him. So please go ahead, Jessica.
Hello. So Dr. Greenbaum is a research track faculty at Tulane University School of Medicine, where he's been working with the Tulane Center for Bioinformatics and Genomics for more than 5 years. He works with all different types of biological data to study genetic factors that are important for the development of disease. Today, he's going to talk about one of his current projects. So if you want to go ahead.
Okay, so thank you for that very nice introduction, and thank you for having me here today. I'm excited to be able to talk to all of you about some of my ongoing research. So if people aren't too familiar with bioinformatics, it's a pretty interdisciplinary research area that combines statistics, machine learning, and molecular biology. And my work mainly focuses on using biological data to study the genetic determinants of osteoporosis and bone metabolism. So today, I'm going to introduce one of my ongoing projects, which is related to the integration of the microbiome and the metabolome, and trying to identify the interactions between these biological factors that might be important for bone health. So when we talk about genetics, I think a lot of people may not realize that we're not just talking about the DNA or the genetic code. Genetics is a pretty big umbrella term that includes a whole bunch of different sub areas that we refer to as different types of omics. And each of these omics provides complementary information that's important for understanding the biological mechanisms that are involved in the development of complex human disease. But the omics factors do not act in isolation. They actually crosstalk and interact at multiple levels, which creates complicated network structures. So integrating the data from multiple types of omics into the same analysis can help us to explore the mechanisms of disease from a systems biology perspective, and can help to uncover valuable insights that we would not learn from studying each of the omics types individually. And our group at Tulane works on developing and applying statistical methodologies for studying the interactions between these different omics factors that might be important for bone health. And we mainly focus on osteoporosis, which is an age-related condition that's associated with reduced bone mineral density and increased susceptibility to low trauma fractures, which are the clinical endpoint. It's the most prevalent metabolic bone disorder, affecting more than 200 million people worldwide. And the burden is especially large among postmenopausal women, which can mainly be attributed to the reduced production of estrogen and other hormonal and metabolic changes that take place in the body during menopause. It's estimated that about 1/3 of postmenopausal women have osteoporosis, and that nearly half of those women will experience fragility fractures in their remaining lifetimes. And BMD is influenced by both genetic and other lifestyle factors, including exercise, nutrition, and aging. The Bmd heritability, or the proportion of the total BMD variation that can be explained by genetic contributions, is estimated to be between 50 and 80%. So BMD has a really large genetic determination. And bone metabolism involves highly specialized cell types, mainly osteoblasts, which are important for bone formation, and osteoclasts, which are primarily involved in bone resorption. And so we typically want to identify biomarkers that influence the activities of these highly specialized bone cells. But despite the extensive genetic studies for BMD, the different omics factors that have been identified so far explain only a small proportion of the total BMD heritability and variation. So there's still a lot to be learned and understood about the molecular mechanisms. And recently, in the past few years, the influence of the microbiome on human health has become a really popular research area. The gut microbiome is composed of the bacteria that reside in the human gastrointestinal tract, including more than 1,000 distinct bacteria species that have been identified so far. And these organisms are really abundant. There's at least a 1-to-1 ratio between the number of microorganisms in the microbiome and the number of human cells in the body. And the metagenome refers to the combination of the unique genomes of all the bacteria species in the microbiome. Collectively, the microbiota contribute about 3 million genes, which is about 100 times larger than the number of genes in the human genome, which is somewhere around 20,000, give or take. So we often refer to the microbiome as our second genome. And the microbiota are involved in a number of diverse processes that are important for human health. For example, they communicate and exchange molecules with the human cells. They contribute to metabolic processes and digestive enzyme activity, and they can interact with the human immune system. And there are several potential mechanisms through which the microbiome may influence bone health, which we previously summarized in our review paper that's published in the Journal of Clinical Endocrinology and Metabolism. Firstly, by interacting with the intestinal epithelial barrier and by altering the pH of the gut, the microbiota can influence the absorption of essential minerals for bone metabolism, like calcium or magnesium or phosphate. Secondly, the microbiota are foreign organisms that live within the body, so they can elicit immune responses either locally in the gut or systemically throughout the body, which can alter the levels of different cytokine proteins, including some that are important for bone health, like TNF-alpha or IL-6. Thirdly, the microbiota produce a variety of metabolic byproducts, which can regulate critical cell signaling pathways that are important for bone remodeling. And lastly, there's a gut-brain axis where the microbiota can regulate the levels of certain hormones and neurotransmitters like serotonin. And it's been shown that bone cells like osteoblasts have serotonin receptors, and higher levels of serotonin have been associated with reduced bone mass. So the microbiome has been linked with a number of risk predictors of osteoporosis, like obesity and diabetes. But only a few limited studies in humans have explored the potential importance of the microbiota for bone health. And these early studies had small sample sizes with no specific bacteria or functional mechanisms that were identified, which is a major limitation. So the adult microbiome composition usually remains pretty stable over time, although it can be altered by a few different factors, like changes in the diet, aging, or medications like antibiotics, or changes in the host immune status. But several population-based studies that were published in high-impact journals like Nature and Science have estimated that these factors collectively only account for about 10 to 20% of the total microbiome diversity. So recently, this has led to the rise of studies exploring the interactions between the microbiome and human genetic factors. Some studies have shown that there's a heritability component of the microbiome. So people who are related and more genetically similar have more similar microbiome compositions regardless of their environment. And genome-wide association studies have identified links between the microbiota and a number of different genes across the human genome. And when they did knockout experiments of these microbiota-associated genes in mice, they observed significant changes of the microbiome composition, which provides some pretty compelling evidence that there's a complex relationship between the microbiome and the human genome. And in our genome, the DNA is organized into chromosomes in the form of chromatin, which is essentially supercoiled DNA. The DNA loops around histone proteins, which are shown by the orange circles, to form chromatin. And when the chromatin is tightly coiled, the DNA is inaccessible for transcription and genes are turned off or inactivated. On the other hand, when the chromatin is loosely coiled, the DNA is accessible and genes can be turned on. And there are a whole bunch of different types of chemical tags that can be added to the DNA, which influence how tightly the chromatin is coiled. And so epigenomics is the study of these biochemical changes to the chromatin structure that regulate the gene expression levels without actually making changes to the DNA code. And since the epigenome can be regulated by environmental factors, the microbiome has been proposed as an important source of influence. And it turns out that the microbiota can actually produce epigenetically active byproducts, which regulate the DNA methylation patterns of human cells. And DNA methylation is one of the most commonly studied chemical tags in the epigenome. So through these epigenetic modifications, the microbiome is actually an important modifier of gene expression in the human genome. Now, metabolomics allows us to profile the intermediates and end products of cellular metabolism, and metabolites are important for a variety of essential cellular processes, like energy production and storage and signal transduction. The underlying foundation of biology tells us that DNA is transcribed to RNA, which is translated to proteins, which serves some biological function that produces metabolites. So metabolites actually represent the downstream expression of other omics factors, and alterations in other omics can be amplified at the metabolomics level, allowing us to detect critical biomarkers and signaling pathways that are closely related to disease risk. Now, it's widely accepted that changes in the diet can lead to shifts in the composition of the microbiome, but probably even more important is the resulting effect on the human metabolome. Our diet includes a variety of different kinds of components or compounds which cannot be broken down by human digestive enzymes, which pass to the gut where they're broken down by the microbiota. And the metabolic byproducts that are generated during these processes can then enter the circulating blood where they may potentially impact human health. One of the most prominent examples is the gut metabolite trimethylamine oxide, or TMAO, which has been associated with the development of atherosclerosis. When we eat certain foods like steak or eggs, which have high levels of the dietary compound phosphatidylcholine, the microbiota produce trimethylamine, which is oxidized by compounds from the liver to form TMAO. And then the TMAO promotes the accumulation of cholesterol and atherosclerosis, which of course are associated with an increased risk of heart attack and stroke. And these mechanisms have been reported in both Nature and New England Journal of Medicine. And it's especially noteworthy that recently a novel therapeutic approach was established for reducing the microbial production of TMAO. And that really represents the first step towards the treatment of cardiovascular disease by targeting the microbiome and its metabolic byproducts. So in our study, we hypothesized that there might be similar undiscovered mechanisms which contribute to the osteoporosis susceptibility. So we performed a novel, systematic multi-omics analysis for BMD using the paired microbiome and metabolite profiles from 500 early postmenopausal Chinese women. Women usually have a pretty drastic reduction in bone mass during menopause. And so the aim of this study is to try to identify some interactions between the microbiome and metabolites that might be contributing to this bone loss during the menopause transition, which might potentially be novel therapeutic targets for the treatment and prevention of osteoporosis. And to our knowledge, this is the first integrative analysis of these biological factors in relation to bone health. So for these 500 women, we performed shotgun metagenomic sequencing of the stool samples to get the microbiome data, liquid chromatography, mass spectrometry, untargeted metabolomics profiling of the serum samples, whole genome sequencing of the blood samples to get the genotype information, and we use DEXA to measure the BMD of the hip. And we focus on hip BMD because it's the most commonly used criteria for clinically diagnosing osteoporosis, and it's also the most powerful known risk predictor of hip fracture. So all the women in this study were between the ages of 40 and 65, with an average age of 53. They had fairly normal BMI levels. They exercised about once per week. The time since menopause ranged between a few months and 9 years, with an average of 2 years. And because sex hormones are important for metabolism in general, and bone metabolism more specifically, we measured the levels of the follicle stimulating hormone and the estradiol in the serum. And all these covariates have significant bivariate associations with BMD. Now, because of some technical aspects of the metagenomic sequencing process, for each bacteria in each subject, we get a relative abundance rather than an absolute abundance. The total number of DNA read counts per subject are highly variable, and they're constrained by the maximum number of reads that the DNA sequencer can provide. So the observed raw abundances and total number of read counts per subject are actually not informative because they only represent a fraction or a random sample of the original DNA content. So the microbiome data are compositional because the relative abundances of all the bacteria species within each subject are proportions which sum to 1. In mathematical terms, we would say that for each subject, the relative abundances are constrained to a p-1 dimensional positive simplex. So this induces strong dependencies among the different bacteria because increasing the abundance of one species implies that there must be a decrease in the abundances of at least one other bacteria. And so regression analysis with compositional covariates has to take into account the intrinsic multivariate nature and the interrelated structure of the data. So the compositionality can typically be addressed by applying a log ratio transformation to the relative abundances. The ratio transformations capture the relationships between the features in the data, and those relationships will be the same whether the data are absolute counts or relative abundance proportions. And taking the log of the ratios makes the data linearly related. So then, in order to assess the association between individual bacteria and a quantitative outcome variable, we consider the linear log contrast model, where Z is an n by p-1 log ratio matrix, whose pth component is the reference component. But in this formulation of the model, as it's presented on the slide, we can only estimate p-1 regression coefficients, because one bacteria is used as the reference component for calculating the log ratio transformations. So the model can be expressed in a more symmetric form by introducing a new regression coefficient for the last bacteria species, which is equal to the negative summation of the regression coefficients of all the other p-1 bacteria. So in this formulation of the model, we can estimate a regression coefficient for every bacteria by imposing a constraint that the sum of the regression coefficients of all the bacteria species is equal to 0. And we do not need to include an intercept term in this constrained regression model because it can be eliminated by centering the response and predictor variables. And then to accommodate the high dimensional setting, where the number of bacteria is larger than the sample size and only a few of the regression coefficients are non-zero, we can introduce an L1 regularization term or a lasso sparsity penalty into the constrained regression model, where lambda is a regularization parameter to be selected by cross-validation. And so we applied this constrained lasso regression model for 672 bacteria species in our data, adjusting for important covariates. And one of the interesting bacteria that we selected was Bacteroides fragilis, which is one of the most abundant bacteria in the microbiome and has been shown to induce activation of the NF-kappa beta signaling, which is a pro-inflammatory signaling pathway that's associated with bone loss. And Bacteroides fragilis has a strong genetic similarity with Bacteroides vulgaris, which we showed to promote bone loss in vivo in an ovary-ectomized mouse model in our previous study, which is currently under review. Ovary-ectomy means that before we did the experiments, we surgically removed the ovaries from the mice, which is the most commonly used mouse model for studying postmenopausal osteoporosis. We observed that the mice that were treated with Bacteroides vulgaris had significantly lower BMD, significantly reduced serum levels of the bone formation marker, osteocalcin, and significantly elevated serum levels of the bone resorption marker, CTX1. And then using bone samples from each of the treatment groups, we did 3D reconstruction of the bone using microcomputed tomography. And we observed that the bone sample from the mice that was treated with Bacteroides vulgaris was had noticeably more deteriorated microarchitecture compared to the bone sample from the control mice. So after selecting some BMD-associated bacteria, we profiled the functional capacity of the microbiome by annotating the microbiota genes to metabolic enzymes to quantify more than 500 known metabolic pathways in the microbiome community. And for each pathway in each subject, we computed a pathway abundance as a sum of the abundances of the microbiome genes that catalyze the reactions in each of the pathways. And then based on partial Spearman correlation analysis, we identified a number of metabolic pathways that were significantly associated with BMD variation. And all of these pathways involve the microbiota producing metabolic byproducts from the digestion of dietary components. So for example, we identified a negative association of the urate biosynthesis pathway, and uric acid has been reported to induce intracellular oxidative stress and inflammatory cytokines, which stimulate bone loss. And on the other hand, we identified a positive association for the methionine biosynthesis pathway, which is an amino acid that's known to reduce bone loss. Now, metabolomic profiling can be performed on various types of biospecimens. Although we did the serum metabolomic profiling in our study, the fecal metabolite profiles are actually the most representative of the direct metabolic output of the microbiome. So next we performed predictive metabolomic profiling to impute the fecal metabolite profiles based on the observed gene abundances in the microbiome community. Elastic net prediction models were trained using independent reference samples with both microbiome and fecal metabolite profiles to learn a set of microbiome genes that are predictive for each gut metabolite. And the imputed metabolite abundances were computed as a linear combination of the microbiome gene abundances using the abundances from our data with weights that were learned from the independent training data. And in this case, the training data included 155 subjects, and prediction models were considered for 80 well-predicted gut metabolites. And well-predicted means that there was a strong correlation between the observed and the imputed metabolite abundances. The BMD-associated metabolites were identified using partial least squares regression, which is well suited for the metabolomics analysis because of the collinearity between functionally related metabolites that are involved in the same biological pathways. PLS decomposes the predictor matrix to a set of uncorrelated components or latent vectors, and then performs least squares regression on these components. So it tries to maximize the covariance between the outcome variable and the projection of the predictor variables. And we can calculate a variable importance and projection, or VIP score, which summarizes the contribution of each metabolite to the model. The VIP scores are calculated as a weighted sum of the squared correlations between the PLS components and the quantitative outcome variable. And we identified several imputed short chain fatty acids, which were identified for positive association with BMD, including butyrate, propionate, and valeric acid. Short-chain fatty acids are exclusively produced by the microbiota through the breakdown of non-digestible dietary fiber. And butyrate has been really well studied, and there's some papers published where they did extensive wet lab experiments to validate the beneficial effects of butyrate on bone formation. But our previous study is actually the first to report that valeric acid influenced the activities of bone cells that were cultured in vitro. We showed that valeric acid inhibited the production of the osteoclast bone resorption cells, and that it promoted the formation of the osteoblast cells, which are important for bone development. And so, although the fecal metabolite profiles are the most representative of the direct metabolic output of the microbiome, the serum metabolite profiles actually provide a window into which gut metabolic byproducts are actually absorbed into the circulating blood. Many of the compounds from the fecal metabolite profiles may actually be excreted from the body without ever influencing human health. So in the serum metabolite profiles, we considered 381 compounds with putatively confirmed identities based on matching the molecular mass and the chemical structures to the authentic standards. And we found that several of the BMD-associated serum metabolites were intricately linked with the microbiome. For example, we identified phenylpropanoic acid, which is a phenolic acid compound that's produced by the microbial digestion of dietary flavonoids. And flavonoids can be acquired from a variety of different food sources, like broccoli, asparagus, and leafy greens, black and green teas, citrus fruits, raspberries, blackberries, and blueberries, and soy foods like soy milk and tofu. And flavonoids have been reported to decrease the risk of a number of age-related conditions, like cardiovascular disease and diabetes. And they have a lot of promise for protecting against bone loss because of their antioxidant properties. So then we performed a co-inertia analysis to investigate the co-structure between the multi-omics data sets, which can be thought of as a principal component analysis of the cross-covariances. The PCA analyses for each data set are performed separately, but the resulting components are constrained so that the covariance between the projected score vectors is maximized. So it simultaneously tries to find ordinations of the two data sets that are similar. So figure A shows the projection of the metabolites colored by their chemical class. And figure B shows the projection of the bacteria colored by their phylogenetic group. The axes correspond to the first two dimensions of the feature space. And features that have a further distance from the origin have a larger covariance with the other omics data set. And metabolites and bacteria that project in the same direction from the origin have positive correlation. Based on the co-inertia analysis, we calculated the RV coefficient, which is a multivariate extension of the squared Pearson correlation. The co-inertia between two hyperspaces is defined as the sum of the squared covariances between all the variable pairs. The coefficient takes values between 0 and 1, where values closer to 1 indicate higher similarity between the paired omics profiles. And we used a permutation-based approach to determine the significance of the RV coefficient. And we observed that there are some statistically significant similarities between the paired omics profiles to be further investigated. So canonical correlation analysis, or CCA, is a frequently used approach for integrating paired data sets. It identifies linear combinations of the variables from each data set which have large correlation. And for our purposes, CCA has some nice advantages compared to regression-based approaches, because it doesn't require us to have any prior biological knowledge about which omics data set is explained or regressed by the other. In some cases, the directionality between omics can be assumed based on the underlying foundations of biology. But in other cases, it's not so clear. For example, in this study, there could be metabolites that are produced by the microbiota, but then on the other hand, there could also be metabolites that stimulate and inhibit the growth of the bacteria. So CCA may be more applicable for exploring the correlations between the paired metagenomic and metabolomic data sets. But a limitation of the conventional CCA model is that it assigns non-zero weights to every feature, which can result in overfitting with a large number of variables. And CCA is also traditionally unsupervised, which means that it identifies correlations between the data sets, but without regard to any disease or outcome variable. So the overfitting issue can be addressed by imposing a lasso sparsity penalty on the weight vectors for constructing the linear combinations, which enables us to incorporate feature selection into the CCA model. The coefficients for those less important variables will shrink to zero so that we identify correlations between a smaller subset of the most important features. And the sparse CCA model can also be extended to be supervised so that we select features that are correlated across the data sets with importance for a quantitative outcome variable. So X and Y represent the paired data sets, and U and V are the canonical variates or the weight vectors for constructing the linear combinations with a lasso sparsity penalty imposed on them. Then we can denote a subset of features Q1 and Q2 from each data set which have a large univariate correlation with the quantitative outcome variable. And the correlation threshold for features to be included in these subsets is treated as a tuning parameter to be selected by cross-validation. And then for any variables that are not included in these Q1 and Q2 subsets, they're automatically assigned zero weights in the CCA model. So this ensures that any variables that are assigned non-zero weights are correlated across the data sets and also have a certain level of correlation with the outcome variable. So I put together a simulation to demonstrate the performance of this regularized CCA approach. And I used a latent variable model to correlate different subsets of features in the data. In each data set, I set some features that were correlated with the other data set and with the outcome variable, some features that were correlated with the other data set but not the outcome variable, and some features that were correlated with the outcome but not the other data set. And any feature, the remaining features that were not involved in these four types of associations were generated as random noise. And in order to make the simulation as realistic as possible, the correlation structures of the bacteria and metabolites in each data set were borrowed from the real data. So the method was assessed based on its true and false positive rates, where a true positive refers to the selection of a feature that's truly associated with both the outcome and the other data modality. So the top row shows the effect of varying the sample size when keeping the variance of the noise at a moderate level relative to the variance of the latent variables. And under these settings, the model did a really good job of selecting the true positive features. With a sample size of 500, like we have in this study, the true positive rate exceeded 95%. And across all of the different sample sizes, the false positive rate was maintained at an extremely low level of 1% or less. The bottom row shows the effect of varying the noise when keeping the sample size constant at 100. And as you would expect, as we introduce more noise into the simulated data, the ability of the model to accurately select the true positives is drastically reduced. But even with high levels of noise, the model still does not select a lot of false positives, which can likely be attributed to the stringency of the lasso penalty. So when we applied the model to the real data, we selected about 20 bacteria and about 15 metabolites that were correlated across the omics with importance for BMD. And the heat map shows the correlation structure between the selected features. And then we can calculate the canonical scores, which are taken to be the linear combinations of the variables within each subject using the weights that were learned from the CCA model. And we were able to show that the microbiome and metabolite canonical scores were significantly correlated with each other, and they were each significantly correlated with BMD. And the CCA selected features were then used as input to construct the Interomics Gaussian graphical model, where the edges represent partial correlations. We selected the top 100 models by estimating a sparse inverse covariance matrix along a path of regularization parameters with the graph lasso penalty to remove the non-significant edges. And then each model was refit without regularization, and we selected the model with the smallest BIC as the optimal partial correlation network. And so this is the Interomics Gaussian graphical model that we constructed, where the red nodes represent metabolites, the blue nodes represent bacteria, and the purple node is BMD. And the edges represent the relationships between nodes when adjusting for all the other nodes in the graph. And we were able to identify the interomics hubs with the highest degree of cross-omics connectivity as the bacteria Lactobacillus delbrueckii and the metabolite phenylpropanoic acid. And we also identified 2 metabolites which were directly connected to BMD. And then the CCA selected features were also used for Bayesian network analysis with structure learning to identify potential interomics causal relationships. Bayesian network analysis relies on conditional dependencies to generate a directed acyclic graph for causal inferences. And we used bootstrap resampling to select the directed connections that were observed in at least 50% of the bootstrap replicates. So here you can see the structure of the directed network that we constructed. And again, the blue nodes are bacteria, the red nodes are metabolites, and the purple node is BMD. And all of the connections in this network are potentially directed from one node to another. And so then we use maximum likelihood estimation to learn the parameters of the network by performing a regression of each node against its parents, where those regression coefficients can be interpreted as the potential causal effects. And there were a number of potential causal directed interomics connections from bacteria to metabolites. And I selected a few of the interesting connections to highlight in more detail. So firstly, we identified that the bacteria Fusobacterium ulcerans may causally upregulate the metabolite deoxycholic acid. And secondly, the bacteria Lactobacillus delbrueckii may causally downregulate the metabolite phenylpropanoic acid. And this connection was also part of a potential interomics causal pathway, which connected directly to BMD. And lastly, we used Mendelian randomization analysis to support the findings from the Bayesian network using single nucleotide polymorphisms or SNPs that were derived from the whole genome sequencing data of the same study subjects as instrumental variables. And Mendelian randomization was proposed to provide unbiased estimates of the causal effects in observational studies without the need for conducting traditional randomized clinical trials. So there's some similarities in the logical structure between clinical trials and Mendelian randomization studies. For example, if we were interested to assess the causal effect of cholesterol level on the incidence of cardiovascular events, we could conduct a traditional clinical trial where we take our study population and randomly allocate half of them to the treatment group and the other half to the control group, and then give the treatment group a cholesterol lowering therapy and follow these people over time and determine if there are any major differences in the cardiovascular events. On the other hand, there are many SNPs across the genome that are associated with cholesterol level. And the genotypes for each of these SNPs are randomly assigned when passed from parents to offspring. And there are two potential versions of each of these SNPs or alleles that we can inherit for each of these SNPs. And so inheriting the allele or the version of this SNP that's associated with lower cholesterol level is analogous to randomly being allocated to the treatment arm at the time of birth. On the other hand, inheriting the other allele that's associated with higher cholesterol level is analogous to randomly being allocated to the usual care arm. And so if the SNP that's under study meets certain assumptions, then the only difference between the groups that are being compared should be their cholesterol levels. So in this way, Mendelian randomization can be thought of as a natural randomized clinical trial that we all enroll in at the time of our birth, whether we like it or not, based on the genotypes that we inherit. And so this is the underlying model for Mendelian randomization. In an observational study, we would like to be able to identify causal associations between a risk factor and an outcome, but we usually can't because the relationship is obscured by confounding factors. So instead, we use a genetic variant as an instrumental variable to assess the causal relationship. And the genetic variant or the SNP can be considered as a valid instrument if it meets three critical assumptions. The SNP must be associated with the risk factor. Secondly, the SNP cannot be associated with confounders. And that's not testable, but it's reasonable. If we assume that mate choice is unrelated to genotype, then the population genotype distribution will be unrelated to confounding factors. And also, our genome can never be changed. The genome that we're born with is the same genome that we have throughout our lifetime, no matter what our behaviors or environmental factors. So it's impossible for confounders to change our genotypes. The third assumption is that the SNP affects the outcome only through the risk factor and not through other independent pathways. And the technical term is that there are no horizontal pleiotropic effects. And this assumption also actually is testable. So for each instrumental variable, we can estimate the causal effect of the risk factor on the outcome by comparing the effect sizes of the SNP on each of them. And in order to have more precision in our estimates, the causal effect estimates from multiple independent instrumental variables can then be combined by meta-analysis. And so these are the same connections that I previously highlighted from the Bayesian network. And we found that all of the connections of interest were supported by Mendelian randomization analysis. And there was a lot of consistency between the causal effect estimates. The last column of the table shows the significance of the intercept term from an MR-Egger regression model, which can be used for evaluating that third assumption that there are no horizontal pleiotropic effects. And since all of these p-values are non-significant, it indicates that that third assumption of the Mendelian randomization analysis is satisfied. And recently, we've actually been performing functional experiments in ovary-ectomized mice to try to validate the potential interomics effects of Lactobacillus delbrueckii and phenylpropanoic acid on BMD. But unfortunately, I don't have any results to show for that today. And so while our study has a number of advantages, it also has a few important limitations. So firstly, among thousands of unique metabolite features that were identified in the metabolomic profiling, we were only able to produce high confidence identifications for a relatively small number. So it's highly likely that some important metabolites were ignored in our analysis. Metabolite identification is still a pretty open research area. Secondly, the methods that we used only consider linear associations. So later, we might use nonlinear extensions to assess the nonlinear relationships, like kernel CCA or distance correlation. And lastly, because this study is pretty novel and there really aren't any other studies to compare with, the findings are difficult to validate through the literature without additional functional mechanistic experiments. And it's also still pretty unclear if the findings can be generalized to other populations. Different ethnicities have pretty different microbiome compositions. And so, or they have different diets and likely very different microbiome compositions. And metabolomic patterns are influenced by a number of factors like age and genetics and menopause status. But I think there's definitely a necessity for more future studies integrating the microbiome and host genetic factors. The microbiome composition can be altered, which makes it an attractive target for therapeutic manipulation. Conceivably, once we understand the interactions between the microbiome and the human genetic factors, the manipulation of the microbiome composition could be optimized for a given host omics profile to reduce disease risk, which would have important implications for the development of personalized and precision medicine initiatives. And so this work is partially supported by several NIH grants. And I'd like to acknowledge my former PhD advisor, Dr. Hongwen Deng, who's the director of the Tulane Center for Biomedical Informatics and Genomics, and the chair of our division at Tulane School of Medicine, and the PI of these NIH grants. And I'd also like to acknowledge our collaborators at Southern Medical University in China, who did the subject recruitment and sample collection, and who have been doing a lot of hard work to help us with the mouse model experiments. And before I finish, I'd also like to introduce a few of the other major focus areas of my research so that you can get an idea for the different types of topics that we work on. So firstly, I'm currently directing the data analysis for what will be the largest whole genome sequencing study for BMD to date, using the whole genome sequencing data from 5,000 subjects, including both Caucasians and African Americans. And it will be the first whole genome sequencing study to investigate the ethnicity-specific differences in the genetic determinants of BMD. Secondly, in the last couple of years, I've been doing a lot of work with single-cell RNA sequencing data, which I guess you could say is sort of the omics technology that's getting the most attention right now. We use the transcriptomic or gene expression profiles of individual cells to do a variety of interesting analyses. For example, we can investigate the heterogeneity of cell types and separate them into different subpopulations. We can explore gene regulatory relationships that are important for cellular development, and we can infer the developmental trajectories of different cell types over time. And all of the early single cell sequencing studies that were conducted in the bone field were conducted in mice. But our group is actually the first to perform single cell sequencing for bone cells that were freshly isolated from human femur bone, which we were able to get from people who are undergoing hip replacement surgeries. And we have a few publications related to this area, including some that are recently published and a couple that are currently under review. Thirdly, I'm currently involved in a project where we're developing a deep learning model using multimodal autoencoders to incorporate the biological interconnections between different omics in order to improve the imputation accuracy in multi-omics data. And we have one recent paper on multi-omics imputation that's published in Frontiers in Genetics. And lastly, I've recently been getting involved in drug repositioning, where we integrate multi-omics and pharmacogenomic data to repurpose already approved FDA drugs based on the genes and signaling pathways that they target. And so by doing this type of analysis, we can try to avoid the long and costly process of new drug development. And as you can imagine, this analysis has become especially important over the course of the last year. And we actually have one paper that was just accepted in the journal Pharmaceutics, where we proposed some potential novel drug combinations for the treatment of COVID. And we also have a few other drug repositioning papers that are either currently under review or in preparation. And so thank you all for having me and for listening to me talk about my research. And now I'm happy to answer any questions.
Thank you very much. That was a great talk.
Thank you.
So what would your next steps be, or your ideal next steps be, aside from what you already talked about that you're doing, like the whole genome sequencing, but in terms of, I don't know if you're looking for drug targets or what's the next study that you would want to do?
So on that last slide that I presented, those are all 4 projects that I'm currently working on right now.
Right.
But I guess really the goal of all of our projects is to try to identify some potential therapeutic targets. I mean, ultimately, we want to be able to take what we learn from the data and then be able to validate them in, either wet lab experiments or mouse models or something, because if we can do that, then that means that maybe it has some ability to transition to the clinical setting, which is ultimately like the goal of all of our research. We want to be able to make a difference in, the medical treatments that people get for the diseases that they have.
Right.
Yeah.
Would a longitudinal study be something that would be worth doing here, so you could see how bone mineral density changes over time and how that's related?
Yeah, that would be really nice, but it's really hard to get longitudinal omics data. So you don't usually see a lot of longitudinal studies in omics research. And if you do, they're usually published in Nature or, some really high impact journal.
What about having the omics, at a single time point like you do, but having longitudinal bone measurements, bone mineral density?
So that would work for certain omics, like the genotype, which never changes. Your genotype is the same throughout your whole lifetime. But other omics, like the microbiome or the metabolites or the epigenome and things like that, they change with environmental factors. And so having the omics at one time point probably wouldn't be that useful for that kind of study. You would need to have the omics profiles at all the different time points.
Right, but I don't know anything about this area, but is it ever used to predict? I mean, could it be helpful in that way to predict if somebody is sort of on a very rapidly progressive trajectory versus not?
Yeah, so yeah, definitely. We can do some prediction stuff, make some prediction models or prediction studies where we try to find biomarkers that are predictive of somebody having, developing osteoporosis later on or something like that. And that's actually an area that we are interested in, although I don't have any current prediction projects going on.
Great. And tell us a minute about your background. What did you do your PhD research in? And just out of interest to the people here, if you don't mind, what was your educational background?
So my undergraduate background, I'll start there, is in human physiology. And so that's where I really got my knowledge about biology. And then I decided to do my master's degree in biostatistics. And when I was doing that, I found out that bioinformatics was, something that existed. And I took a few classes where I got to work with some genetic data and learn some of those techniques for genetic analysis. And I realized that it combined really all of my interests between stats with biology. And so I decided that for my PhD research, I wanted to work on those types of questions. And so I did my PhD at Tulane in biostatistics, and I actually finished about 6 months ago. And then I was able to get a research track faculty position in the division of biomedical informatics and genomics. And so, so now Yeah, basically, this is all the type of research that I work on, working with all these different types of omics data and trying to understand, the interactions that, are occurring between them. And actually, multi-omics integration has some unique statistical challenges because the different omics all have different data types. So they're not always that straightforward to integrate together. And they're usually high dimensional. So the number of variables is almost always larger than the sample size, which means that we always need to be using some kind of, dimension reduction or penalized or regularization methods. So I think it's a challenging research area. And so I really enjoy working on it.
Great, wonderful. All right, well, thank you so much for a really interesting, informative talk. I think we all learned a lot.
Thank you.
Okay, all right, take care. Bye-bye, everyone.